Let me now just say a word about Mark Bhelke, our speaker today. I was at IDT 20 years ago when it was a small company, about 23 years ago, when Mark was convinced to leave the Whitehead Institute at MIT and move over to IDT to head up research and new product development. Over these 20 years, I think with over 40 published, over 40 issued patents, Mark has really overseen the development of literally every new product that's come out from IDT, of specific relevance to today that includes the portfolio of products for NGS, as well as the CRISPR Cas9 system of IDT that I think he's going to speak about now. It is my great privilege to introduce and welcome back to Israel Dr. Mark Bhelke.
I've been involved in development of a whole wide range of technologies while I've been at IDT, the common theme, of course, being that they all revolve around oligonucleotides. But given a bit of a medical background, the areas that I've always had the greatest love for are the things in functional genomics, where synthetic nucleic acids interface with live cells and animals. So that's included antisense, RNAi, and now, of course, CRISPR. And what I'd like to do is begin to review some of the aspects of what we consider to be the right approaches to do genome editing that will have high efficiency and high fidelity. Okay. Well, we're often asked, what's the best way to do CRISPR? Because there's a lot of ways to do it. All you have to have is a bacterial endonuclease, Cas9, CPF1, whatever variant you're working with, and a guide RNA in a cell. And you can achieve that from a wide variety of different approaches. You can express these off of plasmids or viruses. You can express the Cas9 off of an mRNA and put in exogenous guide RNAs. You can put them all in synthetically as a recombinant protein and chemically synthesized RNAs. And there's each one has its own advantages or disadvantages depending upon what you're doing. For example, if you're trying to do whole genome screens very quickly, cheaply, and efficiently, there really is no substitute for the lentiviral approach. That's going to miss some things, so if you end up going into a focused library for smaller screens, then using arrayed synthetic RNAs actually becomes better. If you're going to do precision editing, where you're trying to make a product that's going to matter, where you're going to study it for years, and especially if there's a potential for medical application, you're going to want to do this with the highest efficiency and the lowest likelihood of side effects. And that's going to be done best with the RNP systems, ribonuclear protein with recombinant protein and synthetic oligos. So as was mentioned before, there's more than one approach to make the guide RNAs. For Cas9, the natural system that occurs in bacteria is a two-part system where you have a short CRISPR RNA that has 20 bases of homology to the target region and a short arm that binds to the tracer. The tracer then is a long RNA that binds to Cas9, and the binding of this to Cas9 changes the conformation of Cas9, flipping it into an open configuration, which then is capable of interrogating the genome. We went with working with this system first for several reasons. One, probably the most significant of which is that it's going to be much more cost effective to make reagents out of this kind of a system. The business end of the molecule that conveys specificity is short. And it's very easy for us to make thousands of these short RNAs very economically. The long piece of the RNA, the tracer, we can make in gram scale, which then makes it very cheap to buy. So for economic reasons, it's much more efficient to go with this kind of system. To make it work well in mammalian cells, however, required a fair bit of study to introduce chemical modifications into the RNAs so that they survived and performed appropriately, and we'll get into that in a minute. The other approach is to use a non-natural system wherein both the CR and the tracer are fused into a single molecule. And this was described in 2012 in the seminal paper that came out of Berkeley from the Doudna group. The advantage to this is that in expression systems, you get one molecule of RNA made off a transcription unit. And so much like shRNAs can be made off of a single transcription unit, and then the natural product is a two RNA siRNA. This is similar to that, where you can have an expression system that makes a single long RNA, but again, that's better made off an expression unit. You can make these off of synthetic RNA on chemical synthesis, but it's always going to be more expensive than this, because every time you do this, you create 20 bases of specificity with 80 bases of constant region. And so you're making 100 bases of RNA for every 20 bases of unique content. So there are cases where this is going to be appropriate to use. I'll go into those later, but it's always going to be more expensive. And we have products today that bridge all the different possibilities. So if you're going to put synthetic nucleic acids in the cells, you've got to do a couple of things. And it's very clear from prior history with antisense and RNAi that two critical things need to be addressed first. One is going to be degradation. RNAs, DNAs, everything you put into a cell gets degraded. So you need to have some chemical modification that stabilizes it, because if it doesn't last long enough to do its job, it's not going to work. The other is that all mammalian cells have sensors that are part of the innate immune system. And these work to identify foreign DNAs and RNAs. And for example, most mammalian DNA has a methyl C adjacent to a G residue, and bacterial DNA doesn't. So sensors recognize that and create immune responses. And that was a problem back in DNA issues for antisense. That's also one of the things that triggers immune problems with plasmids. Likewise, RNA has many multiple sensors in there that trigger immune responses as protection for viruses. And if you don't defend against these, you will trigger immune responses and kill your cells. And the body's natural method of evading immune detection is 2'-O-methyl modification. So if you take the 2'-hydroxyl, which in RNA is an OH and DNA is an H, and put a methyl on there, it will evade detection of most of the RNA sensors in the body. And so your own tRNAs, your ribosomal RNAs, these high abundance, highly structured RNAs that otherwise would be very immunogenic are safe. And so you want to put those into the same context into synthetic RNAs that you use for these purposes. So the first things we did when we were developing product lines along this area was to map this out. And so I don't know that if it's visualized here, but this is supposed to be black and this is supposed to be red, because red indicates 2'-O-methyl. From where I'm sitting, I can't really see it, but trust me on that. If you make everything 2'-O-methyl, however, it doesn't work. And why is that? Well, these are structured RNAs that bind proteins, and methylation changes tertiary structure. So you're going to change structure. The other thing is that amino acids make contact points with certain positions on the RNAs, and commonly the 2'-hydroxyl is the site of contact. So if you block that with a methyl, you're going to disrupt the contact. So it's not at all surprising that if you make full 2'-O-methyl modification, that it doesn't work. So we did modification mapping. To make a long story short, you can define which residues in this long RNA are critical for amino acid contact points. In other words, which can be modified and which cannot. And in doing so, we can create maps. So this is a map of looking at the smaller CRRNA. The prior one was the map of the tracer RNA. It doesn't matter what they are. What matters is that we can make these maps that show residues that can't be modified as opposed to residues that can. So for the commercial product that we've launched, the tracer RNA that we have is 58% 2'-O-methyl. This conveys additional stability and helps protect from immune response. The CRRNA is a little bit more complex because the ability to modify RNA without changing structure is influenced by sequence. And with the CRRNA, this 20 bases up here changes every time you change a site. And so we've mapped out on this sequence a pattern that works well, but that may not apply to every sequence. Fortunately, you don't need these things to be highly modified. So we do 58% 2'-O-methyl over in the tracer, but you don't need that for the CR. So what we like to do for the CR is put N modifications that can be universal, and then only go into higher levels of modification internally for those cases where people actually need it. And this again facilitates a one-size-fits-all solution so that it's easier to have high-throughput manufacturing at a reasonable price. So if you're in a setting where you need to have greater nuclease stability, we can help with defining variants that will work well, but again, they have to be customized. Now, the immune side of the equation is really significant, and when we began looking at comparing single guide IVTs with the chemical two-part synthesis with chemical modifications, what we began to notice was a lot of dead cells. So a healthy cell culture looks like this. Cells transfected with the IVTs look like this, and cells transfected with the chemically modified RNAs look like this. And this is a bunch of dead cells. And if you look at these cells for what's happening in gene expression, what you can see is that a splice factor is unchanged between the cultures. But if you look at an interferon response gene, that the cells transfected with the IVTs are 100 to 1,000-fold induction of interferon response genes. So these things are triggering interferon responses, and you may get cells that survive, but you're going to kill a lot of cells and do a lot of damage with interferon activation. So really, you know, just like the lessons learned from antisense and RNAi, you need to use appropriate chemical modification to make use of these safe in mammalian cells. Remember, these RNAs are unmodified in nature, but that's in bacteria where interferon responses don't exist. And this is not news. It's been published. We published with a group from the National Cancer Institute about it, and other groups are publishing about it also. And again, it just makes sense. You know, the nature of RNA doesn't change between RNAi and CRISPR. So while we started out with the two-part system, which we recommend today for most users because it's going to be the most economical, there are other products that we have which will vary in when you need them depending upon what you're doing. And the XT is a version of the CRRNA that's more highly modified. And it has both RNA, 2'-O-methyl RNA, and LNA bases in it, along with some phosphorothioate bonds, so that it's much more nuclease stabilized. So there's going to be settings, just like Ayal earlier today talked about the hematopoietic stem cell situation, where there's higher nuclease content, and the less modified RNAs didn't do as well. This will help. Now, if you want to go further up the spectrum, there are the single guides. And why would a single guide perform really any differently in this regard? Think about it. The greatest attack on RNAs in the mammalian cells comes from exonucleases. Yes, endonucleases do exist, but exos are the primary first stage of attack. And a two-part system has got 4 ends, and a single guide has got 2 ends. There's less RNA to attack with an exon. And so the SgRNAs are inherently more stable, and if you use the en-bloc version, as was first described by Ayal back in 2015, they work amazingly well. We studied much higher modification levels of these and found that for the most part, all that did was increase cost, and it didn't improve performance. So for the basic product that we're selling today, these are the standard design that Ayal published 3 years ago. If people need more highly stabilized versions, we've mapped this all out. We can make more highly stabilized versions, but again, they're not part of a standard product because, let's face it, most people don't need them and they just increase cost for no good benefit. So again, the sgRNA, this is where the artificial part is, where you've joined the CR with the tracer, and you've got the 20 bases then of the target-specific sequence. And if you just simply end modify these, you get almost all the benefit that you need for most applications. Now, when are those applications that you actually need it? If you go into standard cell lines in tissue culture, the CRRNAs and the sgRNAs work identically. And the only thing you're going to do if you get the sgRNA is pay more for it. That's fine with me, but we don't recommend that just because, well, the CRs work fine. But there are cases where they actually do work better. Now, if you start going to low dose, you'll see that the more highly modified RNAs actually have higher potency. Now, RNA is cheap and using higher dose, is fine. But if you're in a setting where you have poor delivery, then the more stabilized compounds will work better. Another is with the hematopoietic stem cells. Again, there are cell types that are going to have richer nuclease environments. And anything with a richer nuclease environment, the more highly modified versions are going to perform better. So here is the case that Ayala has already shown with the RAG2, where in both cases the IVTs don't work as well, but with the two-part system that works fine with RAG2, but with the RAG1 gene, for some reason, the single guide works better. Since in this cell type you don't know which class your gene's going to be, unless you already know, then this is a case where actually spending a little bit more money and getting the SgRNAs makes sense, because your likelihood of success on the first try will be higher. If you're working in standard tissue culture lines, you're just spending extra money for no good reason. Now, where else is it important? And think about it. All of those studies are done with RNP, where you're delivering pre-complex to Cas9, and the second that hits the cells, it's active. If you work with Cas mRNA or Cas coming from a plasmid and then co-transfect in the guide RNAs, that guide RNA has to sit around and wait for the Cas9 to be made. During that waiting period, what's it doing? It's degrading. So if you're using Cas mRNA or Cas from a plasmid, then the more highly modified and structured RNAs like the SgRNAs are going to work better. So again, if you're using mRNA to get your Cas9, go with the SG format. If you're using plasmid and co-transfecting RNAs, do the same thing. But again, for most applications, we don't recommend using plasmid or RNA because they're going to be sloppier, as has been talked about earlier. Now, what about HDR? What can you do to optimize that? We've looked at the efficiency of HDR as you increase distance from the cut site. And what this is it's an experiment to insert an EcoR1 site. So it's 6 base insertion as you walk away from the cut site. And what you can see is right around the cut site, it's actually amazingly efficient. Look at this. We're getting easily up here 20% EcoR1 site addition. And if you really push it, you may be able to get 50, 60%. But as you get further away from the cut site, the efficiency really drops. And it drops faster as you head towards the PAM site than if you head away from the PAM site. Not sure why, but it's a consistent observation. So if you have a choice between different guide sites, choose the one that's closer to wherever you want to fix. Another thing you can do to improve activity is chemically modify your single-stranded DNA template. This is an unmodified ultramer inserting the EcoR1 site compared to ultramers that have some phosphorothioate ends versus phosphorothioate ends plus LNA. Now the biggest boost, your biggest benefit, comes from adding the phosphorothioate residues. And that only makes sense, because this limits the ability of exonucleases to nibble in on the template. And again, remember the hazard that exonucleases represent. Now you can get some more boost by adding LNAs, but on a cost for dollar benefit ratio, the phosphorothioates are cheap and you get most of the benefit from those. The LNAs can give you a little more boost, but they add a lot more cost for only a small benefit. So what we're recommending for people to do now is where you're ordering ultramer type HDR templates to put a couple of phosphorothioate linkages on each end, and that's going to help things. The other thing that we've been looking at is compounds that can help improve the HDR rate. And there's been 20, 30 compounds that have been published that are supposed to improve HDR, and almost all of them don't work. We've tested all kinds of things. So far, we've only found one compound that actually improves HDR, and it improves HDR by reducing the efficiency of NHEJ. And so this is not something which is great for cells. You know, NHEJ is around because your DNA breaks occur all the time in your cells. And if you don't repair them, it's bad. You know, you get genomic instability, cells die, bad things happen. And so the cells don't like to have the NHEJ apparatus messed with. But you can use an HDR enhancer for a short window of time and have relative safety with it. And what we recommend doing is put this stuff in with the medium. If you do an electroporation, zap the cells, and then transfer it to medium with this stuff in it. And let it incubate for 12 to 24 hours and then change the medium. And depending upon the concentration of the reagent, you may start to see some cell death. But you'll see some good results too. And this is what we observe. The blue dot up here represents the total amount of cleavage events we observed. So you can see in things like Jurkat cells with electroporation, we're easily getting 95% editing efficiency. The RNP systems are fantastic. We're getting here up to almost 20%, well, over 20% introduction of an R1 site with the enhancer present. Without the enhancer, 3.5 to 4%. Now what happens is you push the concentration of this, you're going to see no toxicity at this level, but there will be some toxicity here, and if you go up to 50 micromolar, then a lot of the cells are going to die. So what we recommend to people is that they titrate this, you know, and they go up 10, 20, 30, 40, and as you go higher, you're going to see more and more cell death, but you're going to see higher HDR, and you'll get a balance by fiddling with the incubation time from 12 to 24 hours and the concentration of the reagent. So this thing is entirely new. It hasn't launched yet. It's going to be on the market in 2 weeks. So I'm giving people a preview of it now because we've just had so much success with it. The other thing that's nice about this is that because it affects the fundamental repair mechanisms, it doesn't care whether you're using Cas9, CPF1, or whatever. This is boost of HDR seen with the electroporation, with the HDR enhancer with Cas9. Same thing with CPF1. So either any of these methods you use that make a double-stranded DNA break, and then you're looking to get HDR versus NHEJ, it's going to help with. Now, the main thing I want to talk about today relates to off-target effects. And there are two kinds of off-target effects. We've already talked about the immune effects. The other is cutting at the wrong place. And a lot of times people just assume that you're getting cutting at the right place. And we hear all the time from people, like, oh, we looked at the top 5 bioinformatics sites, and none of them had any editing, so it's clean. Well, baloney. That doesn't mean anything. And Ayal talked about it earlier. And this is a lot bigger issue than people want to believe. And why is that? Well, how does bioinformatic prediction work anyway? It's all based on DNA thermodynamics. We know the energetics of binding of A and T and G and C, and we know the destabilizing effects of mismatches. So you can predict the stability of a match-mismatch hybridization event. Now for PCR, the initiating event is hybridization. So a primer binds onto a template, then polymerase comes in, DNA extension starts, and the reaction's going. Does anybody out there use just bioinformatics to ensure PCR specificity? No. Nobody out there does, not one of you. You all use hot start mixes. Why? Because that's insufficient to predict specificity. In CRISPR, it's even worse. It does not start with nucleic acid hybridization. You form a ribonuclear protein complex. This complex goes into the nucleus, and it ping-pongs around looking for PAM sites. When it finds a PAM site, it opens the DNA, and then it sees if you can form a duplex with the guide. And if it likes the structure, it cuts. If it doesn't like the structure, it doesn't cut. And that like, not like, is, we don't know how to predict that. We can find off-target sites with one base of mismatch that have 0 cutting. We can find off-target sites with 4 bases of mismatch that cut at 100%. In fact, we can find off-target sites that cut better than the on-target site. And you know, you can't predict that. You've got to measure that. And this stuff is happening, and it's happening at places you don't expect. So how do you study it? Well, you've got to use some kind of method to empirically measure where off-target effects are happening, and all of these are done by NGS. You can use whole genome sequencing, but I don't think anybody in their right mind wants to spend that kind of money to hunt for these events. Or you can use methods that work with in vitro DNA, circle-seq, site-seq, these kind of things have been published. Or you can go right in the cells. And as Ayal had mentioned earlier, GuideSeq that was developed by the Young Lab at Mass General is the method that we like to use. Now, the original GuideSeq as published had a lot of difficulties, and we've upgraded the protocols. More importantly, we've rewritten the software because the original software missed half of the events. But there's continuing publication of improvements of these methods. And what they do is they measure real CRISPR cut sites by putting a little stuffer fragment in to where cut sites had occurred, and then you use NGS to fish out those sites. Now, these methods are not quantitative, but they are qualitatively able to find most of the at-risk sites. And so what we'd like to do is use GuideSeq, and we do it in a worst case scenario situation. Instead of using RNP, which is very clean, we use it in a cell line that has continuous Cas9 expression. And therefore it accumulates off-target events more frequently. That allows us to find the sites at risk, and then from that we go look for them in the real case scenario, because once we know where to look, then we can find them and quantitatively measure them through more standard NGS approaches. Here's an example. This is a site in the androgen receptor gene. It looked fine to bioinformatics, but there's the on-target, 72 off-target sites, of which that fraction was predicted. Now, that's what's happening. You know, you're getting cutting that's occurring, then you don't know where it's happening, and it's happening. So how do you get rid of that? What are you going to do for it? Well, let's go back to the basics again. This is what happens if you look at Cas9 levels if you compare plasmid with mRNA with RNP protein. So you look and you see at 48 hours, all of your editing is done. All the editing is done that you want to have happen. Anything happening after that is the mischief. And yet with plasmid, you see it's still going up at 72 hours. Huge amounts of Cas9, way more than you need. With mRNA, you get small amounts of Cas9, but it persists longer. With RNP, you get high amounts that rapidly go away, and at 48 hours, it's gone. So all the meat of this reaction's happening in the first 24 hours. And as Ayal says, hit and run. I always like to say it's kind of commando, because it gets in, does its job, and it gets out without leaving a footprint. And in any case, what that extrapolates to is high off-target effects with plasmid, lower with mRNA, lower with protein. Plasmid, mRNA, protein. And here's what happens kinetically. I think it doesn't relate so much to the amount of Cas9, but how long it's around. So here is putting RNA into a Cas9 cell line. So Cas9 is constitutively expressed. The RNAs are synthetic RNAs, and so they get degraded. So the limiting factor here is time in RNA. And the on-target comes up, but then as you add more and more RNA, the off-target reaches the on-target frequency. If you use RNP, I don't care how much RNA you put in, an RNP you put in, you never reach the on-target frequency at this site. And this is not a mass thing, it's a time thing. And that as you have sustained expression of these things, you get a chance to cut, and a chance to cut again and cut again until you finally affect sites that otherwise wouldn't cut. So, worse plasmid, better mRNA, even better RNP, but even using RNP, you still get some off-target cutting. What can you do about it? Well, There's been a number of papers published that describe mutations in Cas9 that improve the specificity. And these were all done through intelligent design. So that people took the crystal structure of Cas9 and then saw where there were amino acid substrate contact points. They mutated these contact points with the theory being that they were going to reduce the binding affinity of Cas9 to the substrate, and therefore, in cases where there was mismatch, there'd be even lower affinity, and it should reduce the off-targeting. So they did that work, and sure enough, it worked. The off-target effects went down, and they got publications in Nature and Science. We thought that this was a very important finding, and so we actually went to try to license the stuff that was designed by Keith Young. And thankfully for us, their lawyers at Mass General were really slow. And we had tested what they did, it worked. But while we were waiting for the lawyers, we started to do RNP walks along genes and realized that's not working. And what we saw was this, that if black is wild type, and the yellowish green stuff here is the mutant protein that was done at MIT by Feng Zhan, and the yellow is the mutant from Keith Young. And what you notice as you start doing RNP walks along a gene is that a lot of these sites, you're not getting any cutting. And what they had developed were highly mutated proteins that indeed had higher specificity, but they were so compromised for activity that they didn't work as RNP. They worked in plasmid overexpression systems that were used in the papers, but they're working very poorly as RNP. And we wanted this for RNP because, let's face it, if you're going to do medical editing, you're not going to put plasmid in. So we developed a new screen. We made a mutant library of 250,000 Cas9 mutants and put it into bacteria that had two plasmids in it. One was a toxin linked to an on-target site. So if you cleaved that, the toxin went away and you survived. So if you had low on-target activity, you didn't cleave the plasmid, you died. The other was an off-target site put into a selection marker, in this case chloramphenicol. So if you did cleave that, you died. So to come through the screen, you had to cleave this site and you couldn't cleave this site. So it's selecting for high on, low off. And flushed 250,000 clones through the system and came up with several hundred that survived. We then changed the sites so that we weren't working with single sites. Did it again, came up with over 100 sites that worked, then converted those to forms that we could test in mammalian cells. And out of that came one mutant that really stood out as being better than the others. And that's now the blue line. So it's not 100% on target. There's some sites where it still doesn't work as well. So maybe 10% of sites don't work as well with this clone. With the other clones, 50 to 90% don't work as well. So this isn't perfect, but it does work. The other thing is, does it limit off-target effects? So here's looking at 3 known off-target effects in 3 genes. And you can see that all of the clones eliminate the off-target effects. But yet, as you vary site to site, this new single mutant that we isolated maintains activity while the others do not. Now here's an example then of putting it to real life use. So let's go back to that nasty site I showed earlier in the androgen receptor. So here's the off-target profiles you get with it if you use the constitutive cell line. If you switch to RNP, look at the improvement you get. Huge improvement just by getting rid of plasmids. Now let's mutate the cell line to become the HiFi. One amino acid change, that's all it is. And you go from this to this. Then if you take the HiFi mutant and use it as RNP, you go to that. And so we've gone from 28% on target to 99% on target. Not perfect. There's still some stuff that sneaks through, but it works. Here's looking at 7 different sites in different genes, same kind of concept, and in all cases, we're dramatically improving the specificity. So at this point in time, Right now, I think the best methods for high specificity editing are to use RNP with the mutant. Now, one of the things that's important to note here is, in these kind of studies, again, we define the sites of off-target risk to look at by guide-seq. That's a cumbersome procedure. It takes a long time to do, and it's non-quantitative. What we then do is find the sites that are at risk, and we make what we call a ramp-seq pool. And I'll talk more about ramp-seq at the NGS talk this afternoon, but basically what it is it's a method to do highly multiplexed amplicon NGS in one tube with very little pain. It's difficult to look at 50, 100, 400 sites all in one tube without getting just a lot of primer dimer garbage. We've got a way to overcome that. We'll talk about that later. But again, the ability to look at these kind of things fast and easy is really best done through amplicon sequencing, which, if you can do highly multiplexed, is far easier and cheaper to do. So what about clinical use? Well, this goes back to Matt Porteous, where and IL described some of the earlier work done. We've continued working with Matt, and they're working again on the correction of the sickle cell mutation. And in the case of sickle cell, there is a PAM site right near the base change that causes the sickle mutant. That guide site, however, has an off-target site which cuts at 20% efficiency. Now, it's not in a gene, but nevertheless, that's at risk for causing structural rearrangements and other problems. And if you're going to submit to the FDA that you're going to do a clinical study, you want to reduce your risk as low as possible. So they were prepared to submit the IND for this as is, but then they tested our mutant. And their on-target rate stayed the same, but their off-target rate dropped 20-fold. And as a result, they're going to be submitting the IND using the HiFi. If you want to read more about that, the development of the HiFi mutant and all the work with sickle cell was recently published in Nature Medicine in August. And this is the paper that came out with Professor Porteous leading the clinical effort, and my lab leading the mutant HiFi effort. Now, to make that useful, if you want to go into clinical studies, you've got to have GMP Cas9. And IDT does not make pharmaceutical grade reagents. That's a different kind of business entirely. We've partnered with Aldevron, and they do. Now, you can go out and contract to have a GMP lot of protein made, and it's going to cost you a whole lot of money, and it's going to take a lot of time. What they've done now is Aldevron has wild-type Cas9 and HiFi Cas9 GMP pharmaceutical grade in stock, so it can be obtained right off the shelf for a clinical trial. So it doesn't matter if you only need 50 milligrams of protein and somebody won't do a contract GMP synthesis for anything less than a gram. Now you can just buy what you need right off the shelf from Aldevron. So this is going to make accessibility of these to small phase 1 trials much easier. So the conclusion of this is that there's many ways to do genome editing. They all have a different place. If you're going to do precision editing, we recommend using RNP methods. And frankly, now that we have the HiFi, the real question, I think, comes into why not use it. Most sites work well, and the reduction of off-target effects is dramatic. And it doesn't really cost all that much different from the regular mutant. So really, I think if you're doing precision editing, the real question has to be why not use HiFi. So I'd like to thank all the people in the lab that did this work. In particular, I'd like to mention Chris. He's our bacterial geneticist who came up with the screen to do all that mutant testing that allowed us to screen 250,000 clones in a week. And without that, we couldn't have done this. So thank you for your attention, and I'll take any questions you may have.
plasmid, like a tet-on, very low expressing one, to get similar levels of protein, but then it's easier to get into the cells.
You know, it's actually as easy, if not easier, to get protein into the cells. Plasmids have a host of other problems. They induce immune responses, and they're at risk for integrating in the genome in non-specific ways. You know, there's just really, plasmids have so many other side effects that people have ignored for years because there wasn't an alternative to do what you wanted to do. And they've become to accept those failings because there wasn't another approach. For this, there is another approach, and it's just, frankly, a whole lot cleaner and more efficient.
For cells, but for in vivo.
For in vivo, I think there's a couple of things. One is going to be there are nanoparticle methods that are developed that can begin to address delivery of RNAs. That's a case where I think Cas9 mRNA may come into play, because you can get the Cas9 in with an mRNA and then the guide as a single guide format. And I think that's going to be perhaps a better way. If you can get plasmid in, you can get other stuff in. I think the greater issue is going to be cells that you can't get anything into, in which case viral methods may be what you have to use. And again, there you're faced with more of the issues of sustained expression. You can try to play games with inducible, which are always leaky, or you could also try inducible but put in some of the HiFi mutations. Now, in that setting, I'm not sure yet which mutation would be best. One of these more highly mutated forms may be good in that setting. We know from this data, well, anyway, remember that thing where I showed you the HiFi versus the wild type cell line, how the HiFi mutation really dramatically reduced off targeting. We know that in this setting that the HiFi mutation works pretty well. We have not compared that in a constitutive expression system to compare the other mutants. And that's something that if we're going to go to viral methods, we'll want to do.
Do you have any feedback about your modified guides and also your HiFi in mouse embryos?
We have people using the HiFi right now in mouse embryos, and the feedback has been very good. After the publication by Genentech showing widespread, you know, high enough frequency other events occurring that it's concerning, interest in that really has picked up quite a bit. And we've got good feedback about that. Both the SgRNAs and the 2 parts work well in mouse embryos. Most of the people are using the 2 parts because they're cheaper, and the mouse embryos are a low nuclease environment. All right, well, I think it's lunchtime, so thank you.